Hello students, I welcome all of you to my class, the zero class, and we have started a series of lectures which will cater the needs of class 11, 12 and NEET students. BSc students preparing for different entrance exams can also be benefited by these lectures. I urge all of you to share these lectures with uh, others on your Facebook page or through YouTube link uh, so that they can reach the target audience. So our first topic is the kingdom Fungi. We will start with kingdom Fungi. It's a diverse kingdom, and uh, herein we will first study the general characters of kingdom Fungi. Then we'll look at the reproduction, and then we'll focus on the classification of fungi. In the subsequent lectures, we'll deal with each class of fungi separately. So this word fungi, it was for the first time coined in 1973 by Carl Linnaeus, and this is asked in many exams. Who coined the word fungus? So you can easily write it. Uh, in 1973, it was coined by Carl Linnaeus. Then this kingdom fungi, it was given as the separate states. It was included as a separate kingdom. And when we were studying five kingdom classification, we saw that there are certain characters that are specific to fungi, and there are some characters which are similar to that of plants, and there are certain characters which are similar to those of animals. So it was necessary to elevate fungi as a separate kingdom. There are more than 100,000 species of fungi. Uh, uh, New species of fungi, they are being discovered every now and then, and there are already more than 1 lakh species that have been identified. Now, the study of fungi is known as mycology. The study of fungi is known as mycology, and it is derived from the Greek word myx, meaning mushroom, and logos, meaning study. So we have mycology, and anybody studying the mycology is known as mycologist. So anybody carrying research on fungi is a mycologist. And Henrik Anton de Barry, he was a German mycologist, is known as father of mycology. And in India, Sir Edwin John Butler is considered as the father of Indian mycology. Now these fungi, they are eukaryotic. They are eukaryotic. They possess all the characteristic features of eukaryotic cells. Then they are multicellular. You will see all fungi, they are multicellular except yeast. Yeast is unicellular. Then they are achlorophyllous. They do not possess chlorophyll. They can possess other pigments which make them colored, and generally it is considered that the colored fungi, they are poisonous, but chlorophyll is absent, so they are heterotrophic. Here I have already mentioned heterotrophic. They cannot prepare their own food. They cannot carry out photosynthesis. They are thalloid. Their body is not differentiated into root, stem, or leaf-like structures. And then they are also spore bearing. They do not produce seeds. They produce spores. They produce specific spores. And then moving to the nutrition, they are heterotrophics. They cannot produce their own food. They are dependent on other organisms for food and nutrition. Now here they can be saprotrophs, living on the dead and decayed material. So examples are rhizops, aspergillus, penicillium, agarix, and morchella. This is here I have included one photograph of rhizomes. This is the bread mold. It grows on the bread. Then I have included, they can also be parasites, for example Candida, Puccinia and Trichophyton. Here I have included one example of cordyceps. It is a fungus. It grows on dead ants, and when it grows on dead ants, due to the growth of this fungus, the ants, the dead ants, they show movement, so they look like those zombies. So these cordyceps, it is parasite on ants. So they can be parasites on animals, they can be parasites on plants as well. Now here I would like to include one more point, and that point is, that they can also show symbiotic association. Symbiotic association. Symbiotic association. They can sometimes form association with algae. Then they are known as lichens. Algae plus fungi. And fungi, the fungal partner, there are two partners in the association. The fungal partner is the most dominant. Then we have also another association that's known as mycorrhiza. It is association between fungi and the roots of higher plants. Okay. So they can also show symbiotic association. Now we'll move ahead. Now we have the body structure of fungi. We have the body structure of fungi. They have a filamentous body, which consists of a slender thread-like structures, which consists of slender thread-like structures, which are known as hyphae. So this part, this is hyphae. This is hyphae. This one, this is hyphae, and network of hyphae. For example, this is hyphae, and if we have a network of hyphae, then it will be called as mycelium. Now the mycelium can be septate or cenocytic. Septate means there are septa present. Each cell is a separate entity. They are separate. So you have septa, you have the partitions between the cells, or you have cenocytic. There are no partitions present. So the mycelium can be septate or it can be aseptate or cenocytic. In cenocytic mycelium, you have all the nuclei in the same protoplasm. The nuclei are distributed throughout the mycelium, but there are no septa. No septa separates the mycelium. So first we have hyphae. These are small thread-like structures. Then we have mycelium, which are network of hyphae, and the hyphae can be septate or cenocytic. Now, fruiting body is the spore-producing organ of the fungus. So when we have the mycelium, it grows on certain substrate, on the dead material. And during the favorable conditions, it produces an aerial structure, and that aerial structure produces spores. This aerial structure is known as the fruiting body. We'll study about it in the coming slides. So fruiting body is the spore producing part of the fungus, and it is aerial. Then the cell wall of fungi is made up of chitin, cellulose or polysaccharides, and the reserve food material is glycogen. Now moving to reproduction, we have three types of reproduction. We have vegetative reproduction, we have asexual reproduction and sexual reproduction. Generally, in case of vegetative and asexual reproduction, there are very minor differences. They only vary on in how they take place. Now, first we will look at the vegetative reproduction. Vegetative reproduction can take place by three ways. First is the fission. Second is the budding. And third is fragmentation. So we have three types of vegetative reproduction taking place in fungi. And in case of fission, a cell divides into two. There is a cell, it divides into two, forming two offsprings, and this usually takes place in case of single celled these fungi, for example yeast. And budding also takes place in case of yeast. You have a cell, and then it produces a small bud-like structure, and then ultimately produces two cells. And both fission and budding, it takes place in yeast. So we have fission yeast and budding yeast. And then fragmentation takes place in case of those fungi which are filaments, which are thread-like. You have the mycelium, they have the mycelium like this. The mycelium breaks down into different parts, and then each part gives rise to the new thallus. So this is how asexual and how vegetative reproduction takes place in case of fungi. Now we have asexual reproduction. Now, as we already discussed it, the body or the thallus of the fungi consists of mycelium, which is the network of the hyphae. Under favorable conditions, the hyphae produces an aerial structure. This aerial structure is known as the fruiting body. And inside this fruiting body, spores are produced, and then these spores give rise to the thallus again. This thallus is haploid. Then it produces a fruiting body, which is again haploid. So from this haploid structure, spores are produced. These spores, because the thallus is haploid, so spores are produced by mitosis. And these spores are again haploid, and on getting these favorable conditions, these spores again germinate into the man thallus. They form the man thallus. Now there are different types of spores that are produced in fungi. First one is the motile and non-motile spores. Motile, a spore, when I say motile, it means it has flagella, it can move, and non-motile, the flagella is absent. When you say flagella is present, when you say motile spores, then we use zoo for them. So they are known as zoospores, motile spores. And when the spores are non-motile, then they are known as aplanospores. First. Now the second type of spores that is produced include endosporic and exosporic. Spores. So here in this case, You have the mycelium endosporic. You have the mycelium, and under favorable conditions, fruiting body is produced. Or I can show it like this. You have the fruiting body. You have the fruiting body like this, and then this fruiting body produce a jacket-like structure like this, a jacket-like structure, and inside this jacket-like structure, spores are produced. This jacket is known as sporangia. It is known as sporangia. It is known as sporangia. So they are produced. The spores are produced inside this jacket. So they are known as endosporic. And in case of exosporic, you have a thallus, then a fruiting body is produced, and the spores are produced on this fruiting body. They are not produced inside the sporangia. So you have endosporic and exosporic. Then there is another type that is known as we have third category of the spores. Third category of the spores, we have the sexual and asexual spores. Sexual and asexual spores. In sexual spores, I will rub this first. Sexual and asexual spores. So we have a spore. A spore is single celled, and then the spore is single celled, and it is haploid. It's M. Now, how this spore is produced? It can be produced by a thallus, by a mycelium, which is M. The mycelium produces the fruiting body, which is again M, and then this gives rise to these spores, develop spores by mitosis, because the fruiting body is haploid, so the spores are generated by mitosis. It is known as, these spores are known as asexual spores. They are produced from haploid thallus by mitosis. Sometimes, under certain conditions, the mycelium is diploid. The mycelium is diploid, 2N, and then this fruiting body produced on this diploid mycelium, which is again 2N, gives rise to the haploid spores N. So here, these spores, they are produced by meiosis. And these type of spores which are produced by meiosis, they are known as sexual spores. So we have asexual spores that are produced from the haploid thallus by mitosis, and we have the sexual spores that are produced from the diploid thallus by meiosis. So we have meiosis in case of sexual spores, and in case of asexual spores, mitosis gives rise to the spores. So this is about the asexual reproduction. We have different types of spores. We discussed them one by one. Now we have sexual reproduction, and there are three these steps in sexual reproduction. Sexual reproduction in fungi, it's very important. Now, in case of fungi, you have this is the mycelium of one fungus. This is the mycelium of another fungus. Both these fungi, they belong to same genus, same species, same genes and species is same, but they can be genetically different. When I say genetically different, that means they can be two different strands belonging to the same genes, same species. Now these mycelium, they are haploid, N and N, and they will give rise to the gamete. This will give rise to the gamete. This will give rise to the gamete. And the gametes will be formed by mitosis because the thallus is already haploid. So meiosis cannot take place. So you have mitosis here, N and N. Then they fertilize. They fertilize to produce a structure like this. Herein, when they fertilize, when they fuse, the protoplasm, only the protoplasm fuses. So first one is known as, this is the first step, which is known as plasmogamy. Plasmogamy, the fusion of the protoplasm. Then this structure which is formed, which is N plus N, it's not 2N, it's N plus N, because the two nuclei, they are separate from each other. They are present inside the same cytoplasm. Only cytoplasm has fused, but the two nuclei, they are separate from each other. Then after some time, this structure is known as dikaryon. It is known as dikaryon. Then after some time, these nuclei, they fuse together. They fuse together, forming a diploid structure. This process is known as karyogamy. Karyo means nucleus. So it is known as karyogamy. So we have a diploid structure now. Now the third step is meiosis. Third step is meiosis. So we have meiosis. This meiosis leads to formation of different spores which are haploid. Now these haploid spores have the potential to give rise to the man thallus again. They have the potential to give rise to the man thallus again, which is again haploid. So these spores, they are produced by meiosis. Here you have the 2N structure. And then you have the formation of spores by meiosis. So they are sexual spores, and on getting favorable conditions, they germinate and give rise to the man thallus again. So this is all about the sexual reproduction. However, in certain fungi, this dikaryotic or dikaryon is not formed. It is formed in those fungi, for example, in case of phycomycetes, wherein plasmogamy is immediately followed by karyogamy. So there is no time for this dikaryotic phase. So you have plasmogamy, you have second is karyogamy, and then meiosis. Meiosis in case of fungi is post-zygotic. After zygote is formed, then meiosis takes place to restore the haploid phase, which gives rise to the mycelium, which is again haploid. Now we'll move to the classification of fungi. So if you see the classification of fungi, it's very easy to remember. I will show you in the next slide. So I'm going to show you the classification of fungi. Now we have two types of fungi in these mycelium. One is known as aseptate or cynocytic. Another is known as septate. In case of septate, you have these septa. The partitions. Now, here you have two types of fungi. The aseptate, wherein you do not have the septa, it's also known as cynocytic. And then you have septate fungi, where septa are present. Now, the septate, all the fungi that have aseptate mycelium, they are included into phycomycetes. They are phycomycetes. And those which have septa, they are categorized into three groups. First group is those which have septa with a simple pore. They have septa, but there is a simple pore present inside the septa. Then these are known as ascomycetes. So based on the presence of spore In case of the septate fungi, when it is simple pore, it is known as ascomycetes. And when you have a structure with a spore like this, a dolly pore, it is known as dolly pore septum. When the pore is not simple, it is a barrel shaped, then it is known as basidiomycetes. And when the pore is present or absent, or there is no information available on the type of pore, whether it is present, whether it is absent, whether it is dolly pore, whether it is simple pore, we have no information, then they are known as deuteromycetes. They are also known as fungi imperfecti. Now, this classification is based on the thallus structure, whether the partitions are present, whether it is aseptate, whether it is septate. So, aseptate, these fungi, they are included into phycomycetes, and septate fungi, they can be either ascomycetes, basidiomycetes, or deuteromycetes. If the pore is simple, they will be called ascomycetes. If there is dolly pore, they will be called as basidiomycetes. If we do not have much information about the pore and septa, then they are included into deuteromycetes, which are also known as fungi imperfecti. There is a reason why these fungi, they are known as fungi imperfecti. Now, this Alex Force was a mycologist. He said that this classification based on the thallus structure is not reliable. So he said those fungi which produce zygospores will be included into this class zygomycetes, and those fungi which produce oospore will be included into oomycetes. Those fungi which produce ascospore will be included into ascomycetes. Those fungi which produce basidiospores will be included into basidiomycetes. These are the sexual spores. These are the spores produced during sexual reproduction. So you use sexual spore as the criteria for classifying the fungi. Now, in case of deuteromycetes, the sexual reproduction is absent. So they are also known as fungi imperfecti. So these fungi included in the deuteromycetes, for them the sexual reproduction is unknown. And if somebody works on a fungi belonging to deuteromycetes and he finds out that this fungi also reproduces sexually, then it is immediately removed from this group and placed on any of these groups to which it actually belongs based on the sexual reproduction. Now you have, in case of zygomycetes and oomycetes, which are the subgroups of these phycomycetes, you have either zoospores, which are motile, and aplanospores, which are non-motile. These are the asexual spores. We first talked about the sexual spores, and now we are talking about the asexual spores. Zoospores and aplanospores, they are produced by zygomycetes and oomycetes. Conidia is produced by ascomycetes, and in basidiomycetes the asexual reproduction is generally present. However, in some species it can be present, and if it is present, then they produce conidia. In deuteromycetes, only conidia are reported. Now, if you see that there is a trend, when we move from lower fungi to higher fungi, the asexual reproduction is either limited to the production of few types of spores, or it is absent. For example, here in case of phycomycetes, zoospores and aplanospores both are produced. In case of ascomycetes, only conidia is present, and in case of basidiomycetes, generally the asexual spores are absent, and if present, then it will be conidia, and then in deuteromycetes you have the conidia. So this is the classification. First, it is based on the mycelium, whether it is septate and aseptate. Then the pore, whether it is simple pore or the dolly pore. And third, the type of sexual spore. And also, when you see this classification, it also follows the criteria of asexual spores. So this is all about fungi. Now in the coming lecture, we will study about the ascomycetes, basidiomycetes, deuteromycetes, as well as phycomycetes separately, and we will see their general characters, and also during that lecture we will discuss about the sponidia, we'll ascospore, basidospore, we'll discuss about all types of spores. Thank you for being with me.